All right. So let's hope it works now when it's me that's on. So yesterday we learned that it was pretty important to be present at all these meetings. This was what would give you prizes, Juan said. But so I just wanted to point out that I started coming to these meetings as soon as I heard about them, which is actually even before 2017. But last year I unfortunately skipped one because I became a father. And as punishment for that, I've said to Anton, my son, when he was 5 years old, you have to read the entire handbook of venomous reptiles, starting with Juan's chapter on venomics. So, but anyway, I'm back again in Oxford, and it's a pleasure to be here. It's always one of these conferences that I enjoy a lot. And as Ronald said, today I will be talking about something different from what I usually talk about. It's been a lot about antivenoms and snake toxins and so on, but a little bit under the radar, at least for the first years. I've also been working a lot with you could say microbial toxins and how to neutralize them. So also maybe I thought, what is the reason why I'm giving this talk in this very sort of snake or animal venom focused group? And it's not only to present the science, it's also a little bit maybe to start a discussion, you could say, not necessarily a scientific one, but a strategic discussion. Because when we read the papers and whatever we hear in the sort of media and amongst scientists is that generally there's a consensus, it seems, that there's not enough innovation, there's not enough investment, decision makers don't care about antivenom. And then I have to say, seen from my point of view, it seems that there's a little bit of skepticism in this community, sort of in toxinology, about collaborating with industry and maybe engaging in startup activities. So my hope with this talk is not only that you learn something scientific, but also that we can maybe start discussing how do we bring some of these different molecules that people are testing, developing, to the next level where we actually start helping patients. And here, I think we need to think beyond just regulatory clinical aspects, but also how do we actually get it funded? How do we get decisions makers really to care about it? So if you didn't catch it in the previous slide, I am a co-founder and therefore also a shareholder of this company, just so you know. And while some of you, there was one person yesterday also said, you're going to talk about something new. Have you switched field? Well, actually, there was just say this is back to life today. Not as big as Amgen, but we share the same building in Copenhagen. Some of you might have seen earlier this month that we raised 30 million euro. This is not just to brag, but this is to exemplify that if you do something that does attract the attention of decision makers and investors, it might bring you substantially more funding than what we can get from the scientific research councils. That is my point with this slide. It is what is needed to bring things out. But anyway, what I was saying was there was someone saying, have you switched field? Are you now started to do something new? No, this started 35 years ago. This is my dad. This is me. My dad was teaching me how to do business plans, as you can see from a young age. You may see that the apple doesn't fall far from the stem. I'm teaching my son to also do stuff that I care about. But approximately 28 or so years later, I had grown up, and my dad said, now you are sort of ready, and maybe we should actually do something together, because he comes with a background as a chemical engineer and knows a lot about manufacturing proteins in very large scale, both for food, feed, and pharma. And I had, at this point, I was finishing up my PhD on how to then discover antibodies, you could say. And from traveling many places and also having had many nasty experiences myself, it was very clear that gastrointestinal infections are really a big issue. They're a big issue to, I think, probably some of you in this room at least once in a while, and they certainly are a major, major issue for infants in places that don't have access to clean water or clean foods. And we probably also are all aware that antimicrobial resistance is a very, very big challenge, especially when we look sort of 20, 30 years into the future, that our antibiotics simply will not work against many of these infections. So me and my dad, we were looking at each other and said, okay, you know how to produce, well, I know how to find molecules, you know how to produce them, but we honestly don't know too much about these microbes. So I was lucky that my old friend, Sandra, was finishing up her PhD in medical microbiology. And just before she sort of finished, I asked her, Sandra, you probably have a lot of job offers because you're a really smart young lady. And she said, yes, she did actually already have some. So please don't take any of them. We should definitely actually do something about this problem, and we should start a company together. And this, you saw the other picture. This was how Back to Life headquarters looked in 2017. There's not a single thing in this table that we actually own. It's all some sort of borrowed, rented, and so on. So it started just with us funding it together with two other guys just privately. But anyway, to get a bit more into the science, what is it that we actually do? And I won't go in this talk too much into our methods. How do we develop antibodies, or in this case, nanobodies? I think we've seen that in some of the previous talks, but more give you some data on how do they actually work? What is their impact when you use them in real systems? So we work in all these different areas, you could say both in animals and in different sort of human segments. But in this talk, I'm going to focus solely on the animals because this is where we have most data and most, you could say, clearly quantitative data. And we are going into pigs. And piglets or pigs are a great model. It's probably the best model for gastrointestinal infections to simulate humans. They have a diet that is not too different from ours, at least compared to some of the many venomous creatures that we hear about at this meeting. They have certainly very different diets than from us. But piglets, of course, receive sow milk when they are small, and when they're weaned, which they are at some point in commercial pig production, they get different infectious diseases, especially gastrointestinal infections, very similar to how human infants also get many infectious disease, gastrointestinal infections when they are weaned. And when you look at it, and I think these are numbers for Europe, it has a relatively high impact, both on piglet health, but also economic impact. This is, of course, partly why financial investors are interested in solving this problem. You can save a lot of money by improving piglet health. So yeah, you see the numbers here. It's about 4 to 8% of the piglets that die due to these infections. They don't gain as much weight if they have diarrhea, basically. So it's a problem worth solving, while at the same time learning something that we might then later apply for the human cases. What we do in Back to Life is that we develop these so-called binding proteins. They're based on nanobodies. They are bivalent. We make bivalent formats just to have the benefit of avidity and also to be able to cross-link different things. They are very easy to manufacture in large scale. We know this because we've done it in 80 cubic meters. This is the standard sort of production size for feed and food proteins. And you can do that at surprisingly low cost. One of the things that's, I think, a little bit unique with our approach is that we're not making pharma products. We're not going for the therapeutic regulatory route. And this is important for two different things that I just would highlight. And it is that it allows you to go into different manufacturing systems that allow for a significantly lower cost of manufacture. And what we believe, or so far what our regulatory advisors tell us, is that it also allows us to put things in food and feed and be, you could say, a first line of defense. That the piglet in this case, or in the future in other products, the human, doesn't need to get the infection before you start applying this. It is something you can use to fortify food, or use similar to how some of you might have tried to take probiotics or yogurts to protect yourself when traveling, as an example. But it doesn't change the fundamental mechanism of action of the molecules. They still are these sort of nanobody-based binding proteins that bind toxins and virulence factors. And in the case of post-weaning diarrhea, it's particularly a pathogenic E. coli called enterotoxigenic E. coli. We've developed sort of a product that combines 3 different binding proteins. One that cross-binds the E. coli bacteria by binding to the adhesins, and then 2 that target some of the important toxins. You don't need to go too much into details with this slide, but the takeaway is when you feed piglets this, you can't see anything. They're just benign proteins. If the piglet doesn't have an infection, they just pass through, slightly digested. You can't see any differences in any of the sort of health parameters. And we can detect the presence of these proteins throughout the gut, and we can subject the proteins to very high temperature. And here you can see it goes up to 70 degrees. You might think, why is that relevant? Because in the therapeutic setting, it's not relevant. But if you go into feed and food, it's really tough processes that your molecules need to go through. So this is simply so you can spray dry it as cheaply as possible. And gastric juice, pH, and bile is, of course, to sort of assess, is it likely going to survive passage through the GI? Here's an example of what one of the proteins does. This is you could say the one that binds the tip adhesins. It's, of course, a little bit hard to see, but by having the bivalent format allows us to sort of aggregate the bacteria. This is of course, in vitro, ex vivo, but we believe the same thing will happen in the intestine. It's a little bit hard to show because it's not the easiest matrix to work in. But at the same time, we also see that the proteins, this is what you see from the growth graph, it doesn't kill the bacteria. It simply just gets them to aggregate. And we speculate that this might help get them through the system quicker. So we're not trying to push the bacteria to become resistant, but simply just get them to stick and not attached to the intestine. So we see it as a little bit as putting roller skates on them, so they just pass through. I think this was a graph showing we can detect their presence throughout the intestine. This was a classic reviewer 2 question. Do you know that they're there? Okay, yes, well, we'll find out. Yes, they are. And of course, another important thing is to understand how broadly neutralizing are these binding proteins, or broadly binding. So we tested them across a number of different isoforms and E. coli strains to see we have a quite good coverage. So going a little bit more into what is the effect of using these, this is an early trial where we have everything very controlled. So we try, so that's not representative for the final application. But what we can see here, and the details of it, you don't need to think too much. It just basically tells that we applied very early after the piglets are weaned, basically the same day, and then for 3 weeks. But what you see is if you do not give these piglets the binding protein and they are challenged actively with an E-tech infection, you have high numbers of bacteria, and you have, you could say, reduced growth, reduced body weight. But if you apply the proteins, especially those that cross-bind, you see a 2-log difference in numbers. So that's like a 99% reduction of the presence of these pathogenic bacteria, and you see approximately 20%. It's not 100. It's not perfectly statistically significant because simply you can only have 30 pigs in a small study, but they gain weight more than the ones that are just being challenged with the bacterium. Another thing that's of course interesting to look at, and here you really don't need to understand the details, you just need to look at the colors. I hardly even understand the details, but do these proteins do anything to other bacteria? A classical chemical question, because everyone using chemistry will know any small compound does something. And when you then argue with reviewer 2, it's a very specific antibody. It cannot bind anything else. They are like, prove it. Okay. So what you see here, you have to look at the different colors. This is, you could say the difference in colors is a sign of how diverse is the microbiome of the piglets. And what we see in the control, where they're only challenged, you see that there's not so much color variation. That's especially in the first sort of week. And this is an indication, it's not a full proof, but it's an indication that there's a disturbance, a dysbiosis in the gut caused by these pathogenic bacteria. So we can discuss this in more detail, but the takeaway message here is that when you give these proteins to a healthy pig, it doesn't change the microbiome composition, and it even protects those that are challenged. You see a stabilization of the healthy. Well, you can't say the healthy, but the microbiome of those pigs that are not challenged. Most importantly is, how does this actually perform in real life? Because despite that these were in vivo studies that I just showed, they are in an artificial environment where we gavage the piglets, where we know what dose of protein they get. We know that they're challenged, but this is not how we envision these proteins will be used in real life. So we did some rather large trials with 6,000 animals, both in Denmark and Spain and the US. And these exact pigs over there are part of this trial. And if we look a bit further into how that went, I think this is the really interesting part of the talk. And here it's important to say, here we added these binding proteins in their feed, and the piglets could eat ad libitum. So we don't know if all of them ate it. Surely some of them didn't eat. Some of them become post-weaning anorexic because they miss their mother and they don't eat for a few days. So it's a very difficult setting out in unclean farms where the piglets kind of self-administer these binding proteins simply just by eating. Also, for you just to get an idea, how does commercial pig production look, at least in Western Europe? You have 4 weeks where they receive sow's milk. That's really short. Then you have this period where they're weaners, which is a further 6 weeks. That's where this trial was done, where they're particularly vulnerable to gastrointestinal infections. And then you have these grower finishers. And I think the most impressive thing here is how quickly they gain weight if they're healthy. So some of the things we looked at when we did this trial was average daily weight gain. And what we could see, especially in the first week, we had a significantly higher, and this was statistically significantly higher, average daily weight gain in the pigs that received feed where these binding proteins were in, compared to farms where they didn't have it. So these were not unhealthy farms. These were just standard farms where some pigs might be unhealthy. And we could also see an improvement of feed conversion ratio. Both of these numbers are, especially the first one is of course a sign of piglet health, but for farmers, this is also a sign of increased profits, basically. Well, the first one is increased profits, the other one is increased savings on food. A very important thing if you want to do anything about the antimicrobial resistance challenge is to look at how many of the piglets needed antibiotics during this trial. And here we were super pleased to see that we saw again, statistically very significant 21% reduction in the use of antibiotics in herds that received this binding protein. And I think it's important also to highlight here that not only is this nice for the piglets themselves, but having animal production is really a huge driver of antibiotic resistance. In Denmark, it's 1/3 of all antibiotics that are used for the first 2 weeks of piglets in farming. 1/3 of all antibiotics in all of Denmark for all diseases, all animals. So reducing antibiotics in piglets, at least in Denmark, really can have a major impact. To sum this up, we sort of see that we expect that this product can sort of be taken into final, well, it is in commercial development. And the nice thing about this compared to many of the other things that are being sold as probiotics or gut stabilizers is we actually know the mechanism of action. And it's really based on deep knowledge generated by many other scientists on how do these pathogens behave, what are the toxins, where do we need to target them, and so on. Yeah, so you could say we've done a lot of work in piglets, and we have multiple programs now running in humans. I think it's not confidential that we obviously also have an E. coli project in humans, plus a few others that maybe I can come and talk about another time. And if any of you are interested, you could say in this field, we recently, or my PhD student, Marcus, wrote a very nice review in Trends in Biotechnology, where where we highlight all of the different examples where people are using single domain antibodies. So there's a few different types of formats to fight gastrointestinal infections, both against viruses and bacteria and parasites. There's of course a big thanks to our primary collaborators, which is the Aarhus University and the Technical University of Denmark, and SEGES, which is a part of sort of the agricultural ministry, sort of say, in Denmark. And then we have a number of unfamiliar funders here, which will be some venture cap people, and then some also more philanthropic people. And now that we've become a bit more corporate, we have a very sort of corporate live better, fluffy title. But I would like you guys to remember that our unofficial mantra is either the good shit or because fecal matters. And with that, I'm open for questions, and we'll just leave this on. If any of you know someone who is looking for a postdoc working in this area with the human pathogens, let me know. And thank you for your attention.